Today, it is my great pleasure to introduce Professor Sarah Hill, who's joining us from the Psychology Department at Texas Christian University. Professor Hill's talk is entitled Cytokines as a Mediator of Condition-Dependent Behavioral Strategies. So please join me in welcoming Professor Hill to Beck.
All right, hi everyone. Thank you so much for having me. And I will, without further ado, go ahead and get things started. Today, I'm going to be talking to you about some research that we've been doing looking at cytokines as being potential mediators of condition-dependent behavioral strategies. So you could have read that on the slide. All right, there we go. So as you all know, within the area of evolutionary psychology, we've evolved from being a field that was primarily concerned with things like human universals. So things like the preference for a low waist to hip ratio, or women's greater preference for partners with resources. And we've sort of graduated into being a field that's able to make predictions about dynamic processes, including things like development and different patterns of behavior. Right, so theories like life history theory, paternal investment theory, risk-sensitive foraging theory, they've allowed us to better understand how and why individuals differ in their patterns of development and behavior. And sort of broadly speaking, these theories predict that organisms are going to make adjustments both to their developmental programs and then also behavior in ways that will help to promote fitness in their local ecological conditions, right? And from these different types of theories, we've been able to make sense of differences between people when it comes to things like pubertal timing, educational attainment, and patterns of risk-taking behavior, right? And just to give an example, within the field of psychology, we've recently seen a lot of research applying insights from life history theory to better understand things like reproductive timing decisions. So for example, we see that researchers have predicted that individual differences in reproductive timing, both in terms of the timing and then frequency of reproduction, should emerge in response to cues in the local environment bearing on the mortality risk and unpredictability in the environment. Right. And so what this research predicts, of course, is that for individual humans who are growing up in more dangerous and resource scarce environments, that we should expect that they might engage in earlier, more frequent reproduction than individuals who are growing up in more benign environments who have the luxury of delaying reproductive goals in the service of first accumulating sort of somatic strength and things like skills that are going to allow them to better be able to compete for resources and compete for mates. And you all are probably familiar with these data points. These are taken from one of Daley and Wilson's classic studies. This was a paper published in 1997, looking at the differences in birth rates based on the amount of violence in different neighborhoods across Chicago. And here they were looking and comparing the birth rates in the neighborhoods, the 10 neighborhoods that had the longest life expectancy, which you can see on the far right-hand column, and then those neighborhoods with the shortest life expectancy, which you can see over there on the left-hand column. And what you can see, the first thing you can see when you just eyeball those two columns is that, the women who are living in the environments that have the shortest life expectancy are having a greater number of children sort of across the board. And the other thing that you can see is that the timing of these reproductive decisions is relatively earlier in the more dangerous neighborhoods than it is in the neighborhoods that have the lowest mortality risk. And in fact, what they find is that the average birth or the average age of first reproductive event in those with the shortest life expectancy is 22.6, whereas the average age at first birth in the neighborhoods with the longest life expectancies is significantly later at 27.3. Right. And so more recently, researchers have sort of taken these ideas from life history theory and sort of shifted the focus away from the outside external environment and the cues to things like immediate resource needs and mortality risk that we get from the outside environment, and shifted that focus inward. And they've been looking at whether or not organisms might also adjust developmental patterns and reproductive decision-making in response to cues in their internal physical environment that have implications for things like their probability of survival, right, and their immediate resource needs. And one model in particular that's been done or created to account for this is what's known as the internal prediction model, or the IPAR model. And according to this theory, or this theoretical model, when individuals are in poor health, which oftentimes tends to co-occur with being in a dangerous and resource scarce environment, because of course that takes a developmental toll on the body. They predicted that when an individual is in poor health, that this should be associated also with expedited reproduction. And the logic here just being the same as it is when we're talking about a person's outside ecology, right? Just this idea that if the risk of mortality is relatively elevated, it makes good sense to begin to reproduce relatively early compared to later, just because later is less of a certainty if your survival probability is lower. And there's been some data that are consistent with this IPAR model. So research finds, for example, there was this really cool paper that David Wainforth did back in 2012, where they looked at people who have chronic illnesses. And what they found is that women who had chronic illnesses were 1.6 times more likely to have a first child by age 30 relative to those who did not have a chronic illness. And just this idea that people might be shifting the timing of their reproductive decisions in ways that would help to facilitate or promote their ability to successfully reproduce before, given that they have a heightened risk of mortality relative to those without a chronic illness. There's also been some evidence bearing on this general idea in non-human animals. There's this cool paper that was published, I think, in PNAS, looking at Tasmanian devils who have what's known as it's called devil facial tumor disease. And if you ever see a picture of this, it's just ghastly. And what they find is that these devils who have this disease, they have 16-fold increase in pubertal timing. And just again, this idea that individuals might be timing developmental events in ways that will help to facilitate successful reproduction, given their heightened mortality risk that they're facing based on internal cues in their body. And so where we were kind of interested in picking up on these ideas was, one, we were interested in the mechanism, right? So if it's true that individuals might be sort of garnering sort of gathering information about the internal condition of their physical body, and then using that information to inform either developmental or behavioral adjustments that should help promote fitness, like what is the mechanism by which they're doing that? And we were also interested in whether or not we could apply these ideas to make predictions about condition-dependent behavioral strategies, right? So the focus of the IPAR model and similar models has primarily been on developmental questions, right? So looking at things like pubertal timing decisions, right, or reproductive timing decisions. And we were interested in whether or not, health-related variables or internal condition-related variables might also play a role in terms of guiding dynamic differences that we see in social behavior that sort of go on in what I always call like real time, right? And turns out that there's a whole bunch of really smart people who've been studying the links between the internal physical condition of the body and then adjustments in behavior for the last 40 or so years. And that is within the field of psychoneuroimmunology, or PNI. Right. And the field of PNI is primarily concerned with looking at neural immune interactions. Right. And what this research finds across sort of all their different subspecializations is that the activities of the immune system, in addition to playing a role in defending and repairing the body and recruiting cells to the site of infection and so on, also plays an important role in helping the brain keep tabs on the physical condition of the body. So much so that researchers, including people like Jonathan Kipnis and people from his research group, have proposed that the immune system, actually we should think of it in terms of all the other ways that we think about it. We should also think about it as being like like a sensory organ, right? And that the job of the immune system, one of the jobs of the immune system should be to keep tabs on how the body is doing and the physical condition of the body, and then alter neurotransmission in a way that is, sort of facilitative in terms of, in their research, they talk specifically about things related to recovery. But as an evolutionary biologist or somebody with an evolutionary biology background, right, it also seems to make really good sense that this could also play an important role in terms of making behavioral adjustments sort of that promote fitness, right? So things that are related to fitness, right? And the primary way that the immune system communicates with the brain is through the activities of signaling proteins such as cytokines, right? And cytokines are signaling proteins that are released by the immune system when it gets stimulated. And it can be stimulated either in response to like acute pathogen presence, but also in response to things like bodily damage. And if you've got cells that need repair and that sort of thing, that also activates immunological signaling within the body. Right. And these, the presence of cytokines play a role in terms of helping the body clear infections and repair, but they also play a role in regulating neurotransmission. And again, primarily the research has been focused on the role of altering neural transmission in a way where you're going to lead to behavioral and psychological adjustments that help to promote healing. And, just to give you sort of a snapshot of some of the research that's been done looking at the role of cytokines in shaping psychological outcomes and behavioral outcomes. Research finds, and there's been a great deal of research on this over the last 40 years in both humans and in non-humans, showing that cytokines play a role in influencing patterns like sickness behavior. This is probably the most classic research that's been done looking at neural immune interactions, and sickness behavior, of course, describes that constellation of unpleasant feelings that we feel when we are infected with a pathogen. And although it was originally believed that feeling lousy when we're sick was something that was caused by the pathogen, it turns out that a lot of the way that we're feeling is actually influenced by the activities of cytokines themselves. They're produced by our own bodies to lead to behavioral changes that will help to conserve energy for the immunological response that's required in order to recover. So things like feeling fatigue, anhedonia, right? These are part of the host response that's coordinated by cytokines to help conserve energy and keep us home, right? So that way our body can repair and defend itself. Right. And since the time that researchers sort of recognized the role of cytokines in promoting sickness behavior, they've since found that they also play a role in influencing things like mood. So we know that a depressed state is something that is caused by cytokine levels. It's one of the causes of depressed mood. They influence perceptions of threat, which evolutionarily we can make predictions about exactly what that looks like, and it's what the data show, and that is that when cytokine levels are elevated, and so your body is undergoing an immunological event, that people tend to experience an exaggerated sense of threat of others and potentially threatening objects. It's also associated with an increased desire for social support and changes in things like learning and memory. Right. And so we know that cytokines play an important role in terms of shaping dynamically changing psychological states. Right. And we also know that there's cytokine receptors all over the place in the brain. And so over here on the right hand side, what you can see is just sort of a side section of the human nervous system. And what it's showing is just the different regions, like here's the cerebral cortex in the top, and then you can see the hippocampus there in the middle has so many cytokine receptors. These are just sites in the human brain that have receptors for cytokines. And this is just simply to illustrate the fact that their presence, or they have the ability to influence just about every key brain region that you could possibly think of, making them a good potential contender in terms of being a mediator of condition-dependent strategies, right? So if our body is figuring out or getting cues from cytokines that's telling the body like what sort of physical condition it's in, right? Given that we know that that cytokines influence neurotransmission, and there's receptors for cytokines all over the place in the brain, these are a good potential contender in terms of things that might influence and be the sort of physiological mediators of condition-dependent strategies. And so what we were interested in is this very question, right? And that is, might the activities of the immune system, in addition to influencing some of these other processes like sickness behavior, right? Which, sort of we know that the goal, right, of sickness behavior is sort of this more immediate, like recovery and defense type of a function. Might they also play an important role in shaping condition-dependent behavioral strategies? And if I can advance my slide, there we go. I'll tell you the question that we started looking at to sort of begin to address these questions, right? And in particular, our sort of broad theory is that we should expect to see that the activities of the immune system, specifically signaling from pro-inflammatory cytokines, which again get released in response to immunological events, that they should play a role in terms of guiding condition-dependent strategies. And we were really interested in first starting out looking at this question by looking at temporal focus, right? And temporal focus was something that was really interesting to us because it's something that we know, sort of based on theory, that should be influenced by a person's mortality risk. Right. So we know, for example, or, just using theory, that, if the risk of mortality is relatively elevated, right, or your immediate resource needs are particularly high, either of these types of conditions should favor a desire for immediate versus delayed resources, because, of course, your probability of survival into the future is relatively lower. Right. And there's been research using a life history perspective on human behaviors that have found evidence for this based on cues in one's external environment bearing on things like immediate resource needs and the mortality risk. And what this research finds is that when individuals are living in or have grown up in environments that are characterized by high resource needs, so things like resource scarcity and an elevated mortality risk, lo and behold, those environments tend to be associated with increases in things like impulsivity, present focus, and a desire for more immediate versus delayed rewards, right? And evolutionarily, all of this stuff makes really good sense because, of course, that means that we're going to hopefully get access to the resources that we need now. We can translate those resources into reproduction and then decrease our probability of dying without first having had the chance to reproduce. So we were interested in taking that research, right? And then again, just shifting the focus inward, right? And so if it's, if inflammation and the presence of pro-inflammatory cytokines are providing in sort of the brain with information about the physical condition of the body, then we should expect to see that cytokine levels should predict a desire for immediate versus delayed rewards. Right. And, in addition to being related to a relatively elevated mortality risk, because if you're somebody with higher levels of inflammation, all else being equal, your probability of survival, right, in terms of like based on the cues you're receiving internally is going to be somewhat lower than somebody who's got lower levels of inflammation. Inflammation is also a context that's associated with elevated resource needs, right? Because generally when inflammatory activity is high, this increases our metabolic needs. And so in addition to, being an internal cue to a heightened mortality risk, it's also an internal cue of elevated resource needs. And both of these contexts should then predict an increase in the desire for immediate versus delayed rewards. Right, so that is our research question, and that's what I'm going to be talking to you about today, is talking to you about some research that we've been doing addressing these questions and whether or not inflammatory activity predicts impulsivity and present focus. So our first our first sort of research foray into this was conducted on a sample of 159 people, and this included 80 women and 79 men. And our recruitment standards for this were pretty strict. So all participants, couldn't have a chronic illness because we wanted just healthy people. They had to be non-obese because that's associated with inflammatory activity. The women could not be on hormonal contraceptives, and we had to measure them during the early follicular phase in their cycle because we wanted to make sure that we had their endogenous levels of sex hormones were kept relatively low, no anti-inflammatory medications, no sex, drugs, smoking, alcohol, or exercise for 48 hours before the study, and they had to do an overnight fast. We had them come into the lab. They did some behavioral tasks assessing present focus or the desire for immediate gratification. And we also gave them survey measures assessing those same things. And then we took some blood from them, which we used for our biological samples. Right, and just to show you first our outcome measures, we had 2 self-report tasks assessing impulsivity and the willingness to delay gratification. And those were the DGI, which is the delaying gratification inventory, and the Barrett impulsiveness scale, or the BIS-11. So those are their sort of self-report of their impulsivity and ability to delay gratification. And then we also did 2 behavioral tasks. The first of which is the temporal focus task. And that task shows participants a series of questions with scales that look like the one that you're looking at the bottom of my screen. And what this is simply a slider scale where you ask participants with a series of different time points, how long from now does 1 week feel? How long from now does a month feel? How long from now does 3 months feel? And then we ask them about that 6 months, a year, 5 years, 10 years, right? And what research finds is that for people who are more present focused, the future feels further away, right? People who are really good at delaying gratification, one of the reasons that they are able to do it is because the future feels really close, right? So to them, they see things as being almost immediately consequential, right? And this has been something that's been pretty well validated in terms of people's actual temporal preferences. And we also did a delay discounting task. And this is the classic delay discounting task where people are asked about their preferences for an immediate versus delayed reward. And the sort of spread between the immediate versus delayed reward sort of varies. And then you look at what a person's hyperbolic discounting function is to determine the degree to which their decision-making or their preferences are biased toward an immediate versus a delayed response. Right, so those are our outcome measures. We also measured inflammatory activity, given that that's our proposed mechanism. And we measured it in 2 different ways, right? The first measure of inflammation is a measure of chronic inflammation, right? And so this you can almost think of as being like a trait, not state. This is a trait-related variable. Right? So when you're looking at chronic inflammatory activity, we're essentially looking at the tendency of these people's white blood cells to respond in an exaggerated way in response to immunological stimulation. And the way that you do that is with an in vitro test. So we took participants' PBMCs, or peripheral blood mononuclear cells, right? We isolated them, we washed them, right? And then we put them into culture with LPS, which is a portion of a bacterial cell wall that causes an immunological response, right? So we then looked at cytokine release in vitro, right, at 2, 24, 48, and 72 hours post-plating with LPS. And essentially what this is getting at is what is the tendency of these participants' white blood cells to respond in an exaggerated way in response to immunological stimulation. And again, we can think of this as being like a trait, where it's something that differs inter-individually, but it's not measuring what a person's body is doing right now, right? We get that measure from our second measure of inflammation, right? Which is you can think of this as being more like a state. Right, so this was looking at what is the participants' levels of inflammation at the time that they actually did the study, right? And this is something that we measure in vivo, right? And so we did this by looking at levels of pro-inflammatory cytokines in the participants' blood at the time of the study. And we also did a total white blood cell count, because this is something that's also associated with elevated inflammatory activity. And with both this and with our in vitro studies, the cytokines that we were looking at were the sort of classic trio of cytokines that are generally characterized as pro-inflammatory, although IL-6 has some anti-inflammatory tendencies, and they behave a little bit differently from one another. And I'll show you some data some new data that we have that shows that they might be doing qualitatively different things with impulsivity as well. But interleukin-6, tumor necrosis factor alpha, and interleukin-1 beta, they're sort of the classic trio of pro-inflammatory cytokines. And then again, like I said, the total white blood cell count. Right, and so we were interested in, here is our hypothesized model, which we studied using latent factor modeling in SEM. And what you can see here is that we're predicting, the first thing to look at is acute inflammation at the top, right? So we're predicting that acute inflammation will predict impulsivity, right? Because acute inflammation is measuring their levels of inflammation at the time of the study. And if it's true, right, that participants or that people, right, if their condition-dependent behavioral strategies are being sort of influenced by cytokine levels, they should be influenced by their levels of cytokines at the time of the study. So that was our predicted path to impulsivity. And then we predicted that chronic inflammation, right, so the body's state would only, trait, gosh, I always mess it up. The body, like as a trait, would only be predictive of impulsivity in as much as it's related to acute inflammation. And this path is important because it's our ability to test the hypothesis that cytokine levels per se, and not just the condition of the body, right? Because a person's cellular tendencies toward inflammation would predict impulsivity directly if it was something else, right? That's actually causing the relationship to impulsivity. Something just to note here, you can see at the top when we look at our latent factor of acute inflammation that interleukin-1 beta is not on there. And this was just because our levels were so low, which is because we had healthy participants. Interleukin-1, you generally can only get detectable levels if you have people who have some sort of chronic illness or sickness, because it exists in very low levels. Although we found a workaround from that, I can talk to any of you at the end of the study about, because we changed how we plate things since the time of the study that allows us to capture more. But we ended up only being able to model acute inflammation using those other 3 variables, just because interleukin-1 levels were so low and we had way too much missing data. And this is what we found. Lo and behold, we found evidence supporting the idea that, to start with, people who have higher levels of chronic inflammation, so those participants whose cells tend to respond in an exaggerated way to immunological stimulation, they also had higher levels of acute inflammation at the time of the study, which is not surprising. And we found evidence that acute inflammation was related to the impulsivity construct. And it's important to note that this impulsivity construct and this path held constant whether we modeled impulsivity as a latent factor like we have it here, or if we divided up, looking at the self-report measures and then the behavioral measures, it still works if we do it that way. If we look at all 4 individually, it works that way too. This is just the most parsimonious description of our model, and is consistent with our results. And what's also interesting here, and is consistent with the idea that cytokines are the mechanism, is that the links between chronic inflammation, which again, we can think of as being a trait variable, right, sort of bearing on a person's physical condition generally, is not related to impulsivity, except in as much as it's related to the activities of acute inflammation. And this ends up being really important for reasons I'll get to in just a moment. Right. We also ran several additional models to test the robustness of the links between inflammatory activity and impulsivity. And our results remained significant when controlling for things like socioeconomic status, age, sex, sleep, stress, exercise, loneliness, BMI, and recent illness. So, I mean, we ran several models controlling for lots of covariates, and the patterns remained. We also ran some alternative models to make sure that what we weren't capturing was actually that, impulsive behaviors tend to be linked to impulsivity or to inflammation, right? So we're predicting that levels of cytokines should predict impulsivity, but there's a lot of impulsive behaviors, things like smoking, drinking, and risky sexual behavior that themselves should be causing inflammatory activity, right? So we ran a reverse model, and we found that the model fit was better for the model that we had proposed than the reverse model. And we also ran an alternative model where we looked at whether smoking, drinking, and risky sexual behavior was is actually leading to inflammation, right? Rather than inflammation leading to impulsivity, then leading to and what we found was that that model didn't work, right? So the behavioral correlates of present focus or impulsivity, smoking, drinking, drugs, or risky sexual behavior. We didn't have drug data in our study. Those variables did not predict inflammatory activity, which lends, again, support for our proposed model and detract in some ways from the alternative. Right. So the results of this study are consistent, with our predictions. Acute inflammatory activity was found to predict impulsivity, and chronic inflammation was found to predict impulsivity only indirectly, which again is consistent with the idea that cytokines might be the mechanism of action here in terms of what is sort of causing this shift. Right, we're currently following up on this with a series of studies, and here's one where we were interested in experimentally manipulating inflammatory levels. So experimentally manipulating inflammation in vivo, and then looking at whether or not we get changes in impulsivity in response to experimentally inducing inflammatory activity. And to test this, we've been doing some studies using the TRIR. So participants in our study, and we collected 158 participants, again, using the same restrictions that we had in our first study in terms of where women are in their cycles and so on and so forth. And participants are randomly assigned to do the TRIR social stress test, which if you all are not familiar with it, it's a situation where participants are told that they have 10 minutes to prepare a speech about why they should get their dream job, and that they're going to be delivering it to a couple of people in a video camera. And then the participants, the research assistant comes in 5 minutes into their preparation time and says, sorry, we're running late. We actually need you to go on right now. And so they bring the person into a room. They're looking at, these unsmiling people in a video camera, and they have to start giving a speech about why they should get their dream job. And then if they don't do it for more, because they're supposed to give this speech for 5 minutes, if their speech doesn't last the full 5 minutes, they're told that they need to keep going. Then once the speech is done, they have to do backward math, and they have to count backwards from something like 1,103 by steps of 11. And if they get it wrong, they have to start over again, right? So this is something that really stresses people out, because most people aren't very good at doing both things. And so that's our TRIR condition. The control condition, people are told to imagine what it would be like giving a speech and doing math. And so they, write all of this stuff down. We then measure levels of inflammation and also cortisol, because we were really interested in, cortisol and inflammatory activity are really tightly linked, because cortisol helps to regulate inflammatory activity. And cortisol is something that's known to be related to present focus or impulsivity. And so we wanted to make sure, especially given that we're trying to prime both inflammation, and then also cortisol in response to stress. We wanted to make sure that the hypothesized links between inflammatory activity and impulsivity hold when you control for cortisol. And so in the study, again, they're randomly assigned to one of these two conditions. We take a biological sample, we measure their impulsivity, and here's what we find. So what you can see here is that participants in the stress condition, right, relative to the control condition, what we see is increases in levels of all three of our pro-inflammatory cytokines. So you get higher levels of interleukin-1, interleukin-6, and TNF-alpha. You can also see that we get higher levels of cortisol in the participants who are in the stress condition, which is exactly what you would expect. And consistent with our hypotheses, we found a positive relationship between levels of interleukin-1 and interleukin-6 on impulsivity or present focus. What we did not expect is that TNF-alpha seems to be doing something very different. And that is that it is actually negatively related to present focus and impulsivity. Now, and we can talk, we're still working our way through that. And we can talk about that at the end of the study, but the overall pattern is consistent with what we found in our original work, with the exception, of course, of TNF-alpha. Importantly, if we do model these things together, they do hold as a latent factor. So together they predict this outcome. And we looked in our first data set just to see whether TNF-alpha was doing the same thing, and it was not. And so we don't know exactly why we have those differences, and we're kind of trying to hash it out. But we also found that all of these relationships between cytokine levels and present focus impulsivity hold when you're controlling for changes in cortisol that occur in response to stress. So again, this is consistent with the idea that we can get these links between cytokine levels and present focus even when controlling for cortisol. One of the reasons that we're really excited about this, and one of the reasons that we included that chronic inflammation piece in our first study, is that in addition to suggesting that cytokine levels might play a role in modulating condition-dependent behavioral strategies, it's also possible that inflammation could be a mechanism that links early life environments to sort of what we can think of as like fast behavioral strategies or fast, behavioral patterns, including present focus, right? And we think that this is a potentially exciting area of research because there's a lot of research indicating that growing up in conditions of low childhood socioeconomic status is linked with chronic inflammation. And there's a whole area within health psychology that's looking at this because it's so robust, it's so frequently occurring. And what this research finds, and I've just got a couple of little snapshots of other people's data down here, is that childhood SES, but not adult socioeconomic status, is related to exaggerated inflammatory activity in response to stimulation. So like that LPS challenge study that we did as our measure of chronic inflammation, what this research finds is that when they do these in vitro challenges, is that people who grew up in low SES or dangerous environments, they tend to exhibit chronic inflammation as measured by this way. And if you look at this bottom left-hand figure, what you can see is differences in levels of interleukin-6, which is one of the cytokines that we looked at in our research as well. What you can see is that those who grew up in conditions of low early life socioeconomic status, they exhibit exaggerated inflammatory activity in response to poly-IC, which is a viral mimetic, and flagellin, which is something that is supposed to mimic a parasite. And what you can see, or a protozoan, and what you can see is that you get this elevated, exaggerated inflammatory activity among those who grew up in conditions of low early life socioeconomic status. And on the right-hand side, you can also see that these individuals also exhibit elevated levels of cortisol sort of across the day. So this was a study where they looked at diurnal rhythms in cortisol among those who grew up in low versus high early life socioeconomic status environments. And what you can see is that at every time point measured, you get a little bit of elevation among those who grew up in conditions that are relatively impoverished, right? And subsequent work by Greg Miller and Edith Chen and their group has looked at this as a potential mechanism behind this exaggerated inflammatory activity, and it seems consistent with this, right? That you get exaggerated glucocorticoid signaling among those who grew up in poorer environments, which then leads to an exaggerated inflammatory response, given that you can get then glucocorticoid insensitivity on the part of cytokines, and that this potentially might be something that's responsible for this exaggerated inflammation in these these folks. And what's interesting for us is that if we're finding, which we have now in a couple of studies, that acute levels of inflammation are predicting some of these like sort of fast behavioral strategies, such as the desire for immediate gratification, one way that we can help to promote sort of long-term planning or other types of behavioral change that we might want to see among those individuals who've been exposed to low early life socioeconomic status could be simply by reducing acute inflammation, right? So given that others have found that early life stress is related to chronic inflammation, and there's a lot of research showing that early life stress is related to present focus, this raises the possibility that if we eliminate this acute inflammation link, right, that we might potentially be able to help promote more long-term planning behaviors among those from early, disadvantaged early life environments, simply by making improvements in health. So, but there's more. So in addition to looking at, our research interest over the last couple of years has been specifically looking at inflammatory activity and its links with impulsivity and present focus and the desire for immediate rewards. But inflammation might play a role in regulating a lot more in terms of individual differences in human behavior. And if you think about it, if you spend some time thinking about what are some of the behaviors that we do that, what the sort of optimal behavioral strategy is should depend on the physical condition of the body, there's a lot. If we can think about things like our ability to cooperate, right? Our ability to sort of trust other people to like return a favor, right? Anything that requires some sort of investment into the future, even learning, right? Because learning is something where you're incurring a cost right now for something that will, give you a benefit later on. Even that might be, that should be something that changes based on the physical condition of the body. And we might expect that you get less investment in the neural processes related to learning in response to inflammatory activity, right? We can use these evolutionary models that are so predictive of so many things. And when we use that and start thinking about that as applied to cytokines and their impact on behavior, there's a lot of things that we can do with it. And indeed, there's been a lot of research in recent years linking inflammatory activity to things like personality traits and different types of personality like syndromes. So we know, for example, that extroversion is associated with higher levels of inflammation, and that openness is associated with lower levels of inflammation, higher levels of inflammation. So not being open is associated with lower levels of inflammation. And same with conscientiousness is associated with lower levels of inflammation. And inflammation isn't just a sick person thing. And as we found in our research, we scrutinized our participants for health just to make sure that, because we didn't want to look at sickness behavior. We wanted to see like what are, these other small differences in our state and the physical health of our bodies, not just like, are you super sick or not, or do you have an autoimmune disease or not? Just like variability in health is associated with variability in cytokine levels between individuals. And that variability may in fact be related to, a number of different types of behavioral outcomes. And just to show you a few, my former PhD student, who's now a postdoc at Baylor, working with Michael Muhlenbein, I can never say that last name. We wrote a paper for this social and personality site Compass, where we sort of projected some ideas about, what are some of the behavioral patterns that we might expect inflammation to be involved with in terms of coordinating behaviors among relatively healthy people. And some of these are things that we have data for, or that other people have data for, not me. And those are things that you can see with the rectangles with solid boxes, and like the one with mating strategies with the dashed boxes. There, nobody has looked at that yet, but it's something that we think is really interesting. But we should expect to see that inflammatory activity, given that it's related to the body's current energy needs, right? Because we know that inflammation increases metabolic demands in the body and is associated with a relatively elevated mortality risk. We should expect to see that elevated levels of cytokines, just sort of like a bare level, Fisher-Price introductory level of predictions, is that we should expect to see that it's associated with psychological and behavioral traits that promote protection and recovery, right? So we might expect it to be related to personality traits that have something to do with those things, right? Emotion, right? And there's evidence showing that cytokine levels are related to anxiety and depression and disgust sensitivity, right? And then in terms of recovery, learning and memory, self-regulation, motivation, so our responsivity to reward, mating strategies, right? We might expect to see lots of different patterns of behavior that are touched by cytokine levels as a way of better matching our behavior, or sort of promoting or encouraging behaviors that would help just, promote fitness given the current state of the physical body. So in closing, the activities of the immune system may play a role in modulating condition-dependent behavioral strategies, right? And they also may play a role in influencing stable individual differences to the extent that some individuals just exhibit elevated levels of inflammation relative to others. So for example, if you're somebody who does have a chronic disease, you are going to have stably higher levels of inflammation than somebody without one, right? And so we should expect to see that that might also be associated with some relatively stable individual differences, in addition to being associated with dynamic behavioral changes in response to changing cytokine levels in response to one's current state, right? And so this can lead to behavioral fine-tuning based on one's physical condition. And it might be a mechanism by which early life stress modulates individual differences in adulthood. So like I said, given that we see that individuals who grew up in sort of harsh and unpredictable environments tend to have more chronic inflammation than those who grow up in more benign environments. And given what we're showing with acute inflammatory activity and it being linked to chronic inflammatory activity, it could be a potential mechanism in terms of making these types of life history adjustments that we tend to see in the literature. And it also might provide insights into other individual differences, both in personality and then also in terms of behavioral choices. So that's it. That's the end. So thank you all. And I'm sorry if I was rushing. I had taught my class right before this, and so my head's a little muddy, but yeah, I guess, Brian, you are going to.
Yeah, no, thank you so much, Sarah. You just hopped the floor here, so that means you're already in the groove. So you didn't need much warming up to get going. So that was great. You've already had your big dump of cortisol in the morning.
Yeah.
So, so that's great. When you were describing your experiment, I just thought of you're imposing academic hell on these people, having to give a talk on short notice and, try to land your dream job. That's, I'm glad I don't live in your catchment area for experimental subjects.
Well, and let me just say one other thing that I didn't say that I think is really cool and that we're really interested in. And I'd love to get all of anybody's thoughts on some of this, but, given that cortisol and cytokines are so, they co-occur because whenever you get cytokine levels increasing, you get cortisol increasing. It's like one follows the other because cortisol plays this role in regulating inflammatory activity. And there are so many things in the literature that, we think are related to cortisol. And I would love to go back. And so we're starting to think about this a little bit in my lab. there's a lot of things that we think cortisol is doing that inflammation might actually be doing. And it would be really interesting to see a greater number of research studies, measuring both things and then, looking at it. And obviously, everything's very complex, and anything that we think cortisol is related to is probably also related to a million other things, but including blood glucose levels. But we're really interested in sort of parsing the effects of cytokines and CORT, given that you can make similar predictions about both sets of molecules, right? Because CORT is sort of, we tend to think of it as being like things that are drawing you to, whatever's happening right now is very salient and important. And we should expect that cytokines have a kind of a similar pattern, but trying to figure out who's doing what, I think, is a really cool and interesting set of research questions.
Yeah, for sure. Absolutely. So I'm going to just sit here looking at the chat window. If anyone would like to ask a question for Professor Hill, feel free to drop it in there. Or you can also raise your hand. I'm scanning the sidebar to see if anyone has raised their hands yet. I see a hand from Jeff Steibel. So the floor is yours, Jeff.
Great. Thank you. And great talk. Super interesting. I almost feel like in hindsight, it shouldn't be surprising, but it is. I'm wondering whether you have a thought as to whether you can tease out or just what your gut is for whether inflammation is driving an impairment to cognitive activity, so neuronal firing versus just cognitive ability. The decision-making itself. And I ask because there's a biologist, his name is Mike Levin, who's been doing a lot of work in animals and some humans showing that cognitive activity is spread throughout the body, not just the brain, which might mean that just in the same way that alcohol or lack of sleep or drugs, impairs activity in some cases and ability at others, it might be that inflammation across the body is doing the same. So we'd love your perspective on that.
Yeah, no, that's really interesting. And I mean, I would think that that's probably part of the picture, right? Because I mean, and they've done studies where they've shot people up with LPS. So people are experiencing an immunological event within their body that's causing inflammation, but they're not actually sick, right? Because their body's not fighting anything, because LPS doesn't actually make you sick, but it does cause inflammatory activity. And what that research generally finds is that you find the same patterns of, sort of like impaired decision-making or like, oversensitivity and all of these other things, even when people aren't actively sick or more sluggish, but their body might be exhibiting these impairments, all of these impairments, including, physical impairments, as a mechanism to sort of conserve energy and get the more immediate resources that are needed to be able to, maintain either or restore health or, sort of maintain the metabolically expensive immunological response. So I think that is probably, so my first answer is like, the research seems to suggest that it's not just like the body is sick and then therefore, not performing the way it should. I actually think that sort of not performing the way that it should is probably part of the mechanism, where the whole body is working together to, sort of become a little lazy and just want immediate rewards and not but all of that is ultimately, evolutionarily for the benefit of getting the more immediate resources to promote fitness.
Interesting. Thank you.
Dan?
Hi, Sarah. Thanks. Really interesting talk. So a couple of comments that might help you to conceptualize the larger picture in which to locate your work, and also maybe to problematize a few of the things that you noted in your review paper. Actually, in a recent paper, Vahid Shakian and Adam Sparks and I, the paper's on sleep, but we're talking about the immune system quite a lot. And we make the point that an innate immunity is fast but dirty, in the sense that there's a lot of collateral damage. And so a fast life history strategy of greater reliance on innate immunity is essentially sacrificing the future for the present. In contrast, acquired immunity takes time to build a library of antibodies. The response is slow. And so greater reliance on acquired immunity over the course of the lifespan should be consistent with a slower life history strategy. And I think that maybe helps to make sense of some aspects of chronic versus acute inflammation in your results. But at the same time, some features of at least the landscape at present are a little bit unclear. So you pointed out the relationship between inflammation and disgust sensitivity, right? Well, and this may be a sampling problem in that maybe the people being studied are not sufficiently, the range of variation is insufficient because they're primarily, university undergraduates being studied for this, right? But if you think about it, someone on a fast life history trajectory, if what I said just now is correct, they should have chronic reliance on innate immunity. So at the level of cytokines, they should display, enhanced pro-inflammatory activity, but they should be much less sensitive to immediate threats, right? Because being sensitive to those immediate threats means missing out on opportunities in the service of caution, right? And that's consistent with going back to the Wilson and Daley work that you cited at the beginning, right? That's consistent with Margot and Martin's whole focus on violence and risk-taking and so on, right? And differences in ecological circumstances that promote that kind of strategy. But in that light, then people with high inflammatory activity should be less disgust sensitive than people with low inflammatory activity, since disgust sensitivity is a precautionary behavior that is only going to pay off over long periods of time. And so here we're maybe seeing the conflict between the acute and the chronic levels of activation. And it could be that a more refined lens like that you're using relative to some of the narrower parameters that have been measured previously might shed light on resolving the paradox of, say, a relationship between inflammation and disgust and similar forms of risk-taking behavior.
Yeah, it's really interesting that you say that because so we found in our research support for the idea of elevated inflammatory activity and suppressed disgust sensitivity. So we actually have a paper that's under review right now where we find that in a couple of different studies. We actually did an aspirin study too, where we randomly assigned people to take an anti-inflammatory aspirin or a placebo. And then we measured their disgust sensitivity. And what you see is you get elevated disgust sensitivity when you give people an aspirin compared to the control group. And so it's very much consistent with this, because the way that we think about disgust, I mean, I know that you share this view, at least based on some of your work. We might have our Venn diagrams might overlap. It's like it's part of the innate immune system. It's this course, sort of that works in concert with our actual innate immune system, where it's this course lens, but it is very costly to be disgusted by things. And if you're a fast life history strategist, right, you can't afford, I mean, disgust is a luxury emotion. You can't afford to be grossed out by a disgusting mate, or you can't afford to be grossed out by, disgusting food, or a disgusting environment. So we also have another paper that's under review right now, making that point that disgust is a luxury. In that you should only expect to see it among individuals who are relatively high in social status. And that you can actually use it as a cue to somebody's social status, because it is so conceptually linked to being sort of in an elevated position. And we find evidence of that too. If you show somebody a vignette and like somebody drops the piece of food and then says, oh, 5-second rule, and eats it, versus like, oh, blah, people assume that the oh, blah person is higher in status and wealth than like conversely. So really interesting. And I think that, I think a lot of, all of this stuff with immunological measures is kind of the Wild West because it's so complex, and there's about 8 million different things that you can measure, and some of it gets at innate immunity and some of it gets at adaptive, and then the immune system has lots of different ways of sort of making up for if it's not really like the T cell response sucks, then you get a different type of response that's making up for it, and so the inconsistencies that we're seeing in the research with respect to immunological parameters and different types of decision making, I think is probably going to end up being a little bit spotty, and people find it and then they find this other thing until we get a little bit more sophisticated in terms of our understanding of the immune system, like generally. But yeah, really interesting points.
So that's great. I look forward to seeing those discussed papers of yours that resolves the tension there in a big way. It also explains, why, as we know, psychologists are higher status than anthropologists, since the anthropologists, an occupational requirement is to have low disgust sensitivity. I see John has his hand up, and I'll yield the floor in just a minute, but I would point out that if the, the context of our current discussion is, proves to be correct, then modifying behavior through the suppression of inflammation isn't as simple as your somewhat off the cuff remark suggested, because if indeed fast life history individuals are more reliant on innate immunity, then you're opening up, now granted, we live in, this zoo-kept environment where a disease is not the same threat as it is for many less privileged people. But by definition, many of the people, even in the United States, who are on a fast life history trajectory do face higher pathogen threat. And if the prescription for, changing their future discounting is to suppress their inflammatory responses, then you're just opening up a new can of worms with regard to greater susceptibility to infection.
Yeah, no, that's super interesting. And, and when we made that, when we were sort of thinking about that, we were thinking about it in terms of our, over sterilized environments that, that we tend to live in the West. But that is actually like a really interesting question, right? Like, and that is like, if let's say that we did an intervention that even like lowers levels of inflammation, like a meditation intervention, for example, and imagine that we give this to inner city kids, like, will we end up with more acute health illnesses, with health problems within that sort of an intervention group relative to what you get if you don't do that sort of an intervention? And that's like a really fascinating question. I always love it when you get something that you think was a really great idea, and then you and then you find out that the law of unintended consequences is going to come in and bite you in the ass. And that could be one of those cases, right, where it would do that very thing. It's really interesting.
Yeah, you might want to connect with Michelle Kraska at UCLA, because she does those mindfulness interventions with at-risk youth in underserved communities.
All right, awesome. I love it.
To treat anxiety, depression, and so on.
So interesting. So interesting.
Okay, John, the floor is yours.
Hi, Sarah. Great talk. And for someone who was recently diagnosed with a chronic inflammatory neurological disease, I've certainly been monitoring my own changes of mood and things like this associated with it. And one of the things that struck me in your presentation was the relationship with threat assessment. And I certainly have been feeling a little bit more vulnerable, but I noticed that your threat assessment wasn't in your last figure, in your larger conceptual model. You had everything out, but you didn't mention threat assessment. And I wondered how you might incorporate threat assessment into, or how would you, seems to me they're good instruments for measuring threat assessment differences.
So other people have done that in their research. And so we have not, our primary focus has been just looking at right now, at least at the impulsivity piece. But other people have done that, like in the way that they generally do it is they'll show, for example, faces of people and just ask them about like how much anger they have and that sort of thing. And then they'll also measure subjective threateningness, like how threatened they feel. And what you see is that people will, when inflammation is high, that they perceive other people as being more angry and having these types of threatening emotions relative to, and they feel more subjectively threatened when inflammation is relatively high compared to when it's low. And these studies, I'm pretty sure, it's been a hot minute since I've read the primary literature looking at that specific effect. but I think if my memory serves me correctly, that they experimentally induced inflammatory activity by shooting people up with LPS or a control. And they found that specific effect. And I think that's like super interesting. Like, and I don't know about any of the rest of you, but and I don't know about your experiences now that you have this inflammatory condition, but when I got my COVID vaccine, and I got the one-shot Johnson & Johnson thing, and I had COVID. And so my immune response, of course, went off the charts, and I felt terrible when I got my vaccination. And, but the first thing that happened to me, because I went back to my office and I started working, was first I felt hopeless. Like all of a sudden I was working and I just started feeling really hopeless. And then I was feeling socially anxious. Like, and I was like, I am having an immunological, and then about 2 hours later, my body started, feeling terrible and everything else. But it was so interesting, as somebody who's been looking at these relationships and research, just to monitor my own sort of psychological responses to it. And it really is very striking and interesting.
Well, not to take up the rest of this time with personal stories of diseases, but you'll probably appreciate mine. I volunteered and was in the Johnson & Johnson phase 3 trials. So I was immunized, I found out because I was unblinded later on December 1st. That was, months before other vaccines were released yet for emergency approval. I happened then to also get a seasonal flu vaccine. And 2.5 weeks later, I was in a hospital, full facial paralysis, hands felt like they were burned. So I was diagnosed with Guillain-Barré syndrome. And of course, we now know that the Johnson & Johnson vaccine is associated with Guillain-Barré and a rare variant of that, which includes facial paralysis, which I had. I mean, I was, they said, about a day away from a feeding tube and a ventilator. I was already on soft diet because I couldn't manipulate food. And I think it was because I got the Johnson & Johnson, which pumped up my immune system. And then the flu vaccine just put it right over the top that it was, primed by the Johnson & Johnson, triggered by the flu vaccine. But I now have chronic version of that. And so in my thinking of threat assessment, I'm not, I assumed it's because I also haven't recovered a normal walking gait yet. Because I have chronic inflammatory demyelinating polyneuropathy, right? So anyway, so it's demyelination of a myelin sheeting of my nerves outside the central nervous system. Thank goodness I don't have MS.
Yeah, oh my gosh, that's terrible.
Well, I'm walking to class, and the diagnosis was most people can do that in 5 years, and I did it in 5 months, but it's still but part of the other, second part of my question actually is kind of similar to Dan's comment about interventions and what that might do, the cost of that in terms of in his comment related to disease. But you did mention that perhaps developmentally an intervention might direct people away from present focusing, and of course if you do that, you're also minimizing their threat assessment, and some of them may be living in dangerous neighborhoods.
Right. Yeah, no, exactly. It's one of those things where you have to really think about the ecological context in which you would, recommend such a intervention, and then thinking about whether the benefits that you would get from, for example, longer term decision making and that sort of thing, whether that would be outweighed by the costs, like the ones that you and then Dan also outlined, which I think is really interesting. Another possibility then for an intervention, as you were talking, is there's a work by I think it's Chris Kazawa and Thomas McDade looking at innate versus adaptive immunological trade-offs based on access to resources during childhood. And so, another possibility is that you just have an earlier intervention, because their research suggests that if you're in an environment with a relatively elevated pathogen load and access to good resources, you're going to, invest more in adaptive immunity, which is generally associated with a lower inflammatory or an anti-inflammatory profile. since you're not relying on this like really coarse form of immunological activity. And so like another possibility is that you like invest more in developmental programs to encourage, like get access to resources and stuff to children when they're developing to change their immunological profile. But then again, it gets into the same situation, right? Where if then they're in the hood and they're not paying attention to the, stuff, do they end up in trouble? And there's that possibility too. So it's very complex. I think
I don't know if I'm going to be able to do field work again because I have a now compromised immune system. But one of the, I happen to, my field site is that environment where you have a high pathogen load and high resources. So it would be interesting to think about what kind of work could be done.
Right. Well, and that would be really interesting. They think that our immune systems that like people living in like the US and so forth, when you're growing up in a like benign home, where you have access to the types of resources most of us have access to, that we might be developing similarly because we get so many immunizations when we're kids, that this might be programming our immune system like as if we were living in, near the equator essentially, and leading us really biased toward adaptive immunity, which some have suggested might be playing a role in the development of allergies and some other things. So
if I could just jump in very quickly, and just an editorial note, because I'm mindful that this is public information, and we all support John in his recovery and are both amazed and proud at the progress that he's making. I'll just point out that rates of Guillain-Barré in conjunction with the COVID-19 vaccines are relatively low. So on the order of 1 in 20,000 at the high end. And thus, you certainly wouldn't want to discourage people from getting vaccinated against.
No. And if I could add to that, boy, am I a pro-vaxxer. So the odds of you getting Guillain-Barré from COVID is magnitudes higher than getting it from a COVID vaccine. And the same thing goes with the seasonal flu vaccine, which is also known to trigger this, DTaP is known to trigger this. There are vaccines that are referred to as on the table, so already acknowledged. But boy, vaccines are wonderful things. One of the things that I've thought about vaccines, and I hadn't thought about that our kids get so many of them, they're getting some hallmark of lots of serious diseases coming their way. But it also, they also intervene in a non-complete way. And so I've wondered how much of our immune systems have atrophied functions because they're not ever developed or tested, because bodies aren't fully fighting off those diseases, right? And I suspect that there's a wider range of function that the body does. And if some of the inflammatory problems we have now is a byproduct of that artificial presentation of disease.
Yeah, that's really interesting. That's a really interesting idea.
Yeah, that's really interesting, John, to think about the timescale at which these consequences arise. I have a question here from Delaney.
I put it in the chat, but I thought I would just ask it face to face. But I was wondering if you could talk a little bit more about the reverse models that you had described, because I just never heard that before, and I wasn't sure if it was just like fully you're testing it backwards or what that meant, and if you could parse that different as like controlling for the behaviors and the expectations you have.
Right. And let me say that we ran, because the reviewers wanted to see so many different models, we've run just about every model you can think of. The first reverse model, where we were just looking at the fit statistic, we looked at whether impulsivity, right? So impulsivity as the predictor, predicting acute inflammation, which then predicts chronic inflammation. So just flipping the order of things and then looking at model fit. But we also did something where we looked at, and I'm gonna draw it for you because I find that this stuff, it's always so much easier in a drawing. So we looked at impulsivity, and then it predicting basically inflammation promoting behaviors. All right, so I'm just gonna call these behaviors, and then those things predicting acute, and then predicting chronic. Okay, and then we also ran it like flipping over. Okay, so here is impulsivity, right? So impulsivity does in fact predict these risky behaviors, right? So we looked at smoking, drinking, and risky sexual behavior. And sure enough, it does, right? If you're impulsive, you tend to do more of these things. These behaviors were not linked to chronic or acute inflammation. So we modeled it like with acute and then chronic, and we also swapped the order and did chronic and then acute, and regardless, it didn't work. And so what this is showing is that it suggests that the relationship that we're finding linking impulsivity to, you know, an inflammation predictor is probably not sort of this, you know, an artifact of the fact that people who are impulsive are also more likely to do things like smoke, drink, and engage in risky sexual behavior, which they were in our sample. But we did not find evidence of those behaviors themselves being linked to inflammatory activity measured either as acute or chronic. I hope that helped.
Yeah, great. That makes sense. And then what about the model fits for the, I guess I was still confused of when you did it backwards, the model fits were then not.
They weren't, the model fit was better when we ran it the way that the hypothesized model. When we ran the reverse order model, the model fit was poorer. And that again is just more evidence suggesting that, you know, obviously not demonstrating causation because we did not do that. But it's more consistent with our sort of hypothesis than it is consistent with the alternative.
Great, thank you.
Yeah, thanks, Sarah. I don't see another hand up or a chat, so I thought I'd ask a question of my own that's arising out of some of my own interests in spatial behavior. And the question I had would be whether you'd be, you know, willing or, you know, extremely resistant to thinking about how, for example, variability in cytokines through this model of its impact on impulsivity might be influencing people's propensity to either stay homebound or to go exploring for resources, or if there's, you know, multiple, you know, counteracting influences where you wouldn't expect to see any change in spatial behavior. But I'm asking this also thinking that, you know, if you were to really try to measure the behavioral consequences of some of these some of the cytokines, you might do so with non-human models, and usually you can only measure a few things in those models, and one of them is how exploratory they are. You know, if you were to, you know, inject one of these cytokines into a rodent, you might see differences in you know, spatial behavior in such a lab context. So are you, number one would be, are you aware of any work that's looked at this in non-human models to see if there is an impact on spatial behavior? And then two, is it anything that you're interested in measuring or examining with your experimental work in humans?
So the answer to two is sure, yes, because I'm interested in everything. The relationship between these things is one that I am all but certain is going to sort of fall in the dreaded inverted U-shaped function, right? Where you know, we know from research on sickness behavior, and there's been so much research on sickness behavior, so I would look into that literature if I were you. And what you find when you inject animals with LPS and they experience inflammatory activity, they don't want to go out. They don't want to forage. Their foraging range becomes smaller. And all of this makes, you know, really good sense when we think about what we want inflammation to be doing. And if you're exhibiting like high levels of inflammation, that suggests that there's sickness and your energy needs are higher, and you should maintain your resources and not engage in sort of goal-directed behaviors. Instead, you should be focusing your energy inward. And there's a ton of research showing that with animals. In fact, part of the sickness behavior constellation is essentially lethargy, right? And so you get that. But when you get intermediately high levels of cytokines, I would expect the opposite, right? So when you get people who don't have sick levels of cytokines, right? You just have people who have elevated normal levels of cytokines, which is something that's consistent with sort of what we see in our sample, where we have healthy people and just look at their basal inflammatory activity. In that context, I think you're going to get high levels of sort of externalizing, goal-seeking behaviors, and then really low levels of cytokines, you get less of that. And so I think, ultimately, if we plotted things out, it would be that, the dreaded, inverted U. So it depends on like how much inflammatory activity you want to measure. And actually this would be a really interesting study, because given what they find with sickness behavior, and that animals don't want to wander, they don't want to go out and explore when you shoot them up with LPS and they're having this really pronounced immune response, kind of like the one we all got when we got our vaccines, right? We felt like shit, we want to go home and lay down, and same with animals. But if we just look at small elevations, like we would get from the Trier social stress test, or what we get, just basally, based on fluctuating, people's individual differences in inflammatory activity, I would expect the opposite, just because if it is something that is promoting, you know what? I'm refining my prediction on this. I think you'd get a straight relationship. And so, it's been looked at in the animal literature. I'm sorry, guys, I just like wasted 5 minutes of time, like kind of walking through, and I'm like, nope.
That's all right.
Well, because I think that it's like it increases your desire for immediate resources, but it makes you less willing to want to work for them, which is consistent with the idea that you're conserving energy, and don't really want to engage in anything that's going to take up too much of a metabolic toll on your body. So certainly that's consistent with sickness behavior. And it'd be interesting. I don't know that anybody's looked at that with sort of normal fluctuating levels of cytokines. And I think it's an interesting question.
I'm going to jump in and make you wish that you'd stuck with your original formulation. I'm not sure that I'm right about this, but listening to this conversation, I asked the Google, and the Google tells me that in rats, there's a periovulatory increase in GM-CSF, pretty dramatic, and day range and locomotion behavior in most mammals, including rodents, including rats, increases around ovulation. So the opposite to the prediction that that a linear model would suggest, possibly consistent with a U-shaped, I don't know, but certainly not consistent with a linear.
It would be really interesting to look at that, and then also look at different cytokines specifically too, because I can imagine that they could do different things, just given some of the idiosyncrasies that they have when you're measuring them. Need a nonlinear model.
Yeah, curvy models are cool. So that's something you could definitely examine in a competing hypothesis way. So do we have any other questions coming from our audience? And if we don't, then I think what we can do is just call this a slightly earlier Beck. And thank you very much, Sarah, for your time here. And it's been a pleasure to see about all the work that you're doing on these fascinating topics. And thanks for bringing in your perspective. And it's given us all a lot to think about. So I'm certainly going to be following up on some of the work you mentioned about sickness behavior, and so will the rest of us on so much you explained to us today. So have a great rest of your day. And everyone, if you could just unmute and thank Sarah again for joining us, and look forward to seeing how this work develops in the future.
All right, thanks everyone.
Yeah, thank you.
Bye-bye.
Thanks, Sarah.